There's a federally threatened snake living in the wetlands just south of Sacramento, and until recently, scientists couldn't find it even when they knew it was there. Welcome to Conservation Science Today. I'm Tucker McPherson, and in this episode, we're talking about giant garter snake, environmental DNA, and a study that's changing how we monitor species we can barely see. The giant garter snake, Thamnophis gigas, is the largest garter snake in North America and is found only in California's Central Valley. It lives in tule marshes, irrigation canals, and seasonal wetlands. Habitats that have been reduced to less than 5% of their historic extent due to agricultural conversion. The snake is listed as threatened under the Endangered Species Act, but because it's aquatic, cryptic, and present at very low densities, standard trapping surveys often come back empty, even at sites where the animal is known to occur. That's the problem a team of researchers from West Sacramento set out to solve in a 2019 study published PLOS One. The solution they turned to is called environmental DNA, or eDNA. Every living organism, fish, frogs, snakes, shed biological material into its surroundings, constantly. Skin cells, mucus, feces, even just molecules floating off its body. In aquatic environments, that material disperses through the water column and can persist for days to weeks. Scientists can collect a water sample, filter out those microscopic traces, and use laboratory techniques to identify which species left them behind, without ever seeing, catching, or disturbing the animal itself. As Schwartz and colleagues explained in their foundational 2017 review, eDNA methods are especially powerful for rare or cryptic species where conventional detection is unreliable. Schumer and colleagues designed a species-specific quantitative PCR assay, qPCR, targeting a region of the giant garter snake's mitochondrial genome called cytochrome B. qPCR works by amplifying target DNA exponentially and measuring how quickly it accumulates. A faster buildup means more snake DNA was present in the sample. The researchers first validated their assay in the lab, confirming it would detect snake DNA at concentrations as low as 0.00002 nanograms per microliter, without cross-reacting, with closely related Thamnophis species sharing the same habitat. In the field, they collected water samples at 52 sites across the southern portion of the snake's range, where standard trapping had failed to detect the species. qPCR was the right tool here, precisely because the snake is so rare and hard to catch. A method that requires physical capture would simply return negative results, and managers would have no information at all. The results were striking. eDNA detected giant garter snake presence at 28 of the 52 sites, more than half, including locations where trapping had found nothing. The assay reliably detected snake DNA in the water samples collected up to 300 meters downstream from a known population, demonstrating the method's sensitivity under real field conditions. For California wildlife managers, this matters enormously. The Central Valley has large areas that have never been comprehensively surveyed, partly because trapping is labor-intensive and can only be conducted when water temperatures are warm enough for snakes to be active. eDNA sampling is faster, cheaper, and can cover far more ground. The authors did note one important limitation. The method confirmed presence but couldn't estimate population size or density. So eDNA works best as a first-pass detection tool, not a replacement for population monitoring. Still, for a species whose distribution is still poorly mapped and whose habitat is still being lost, knowing where it actually is may be the most urgent conservation question of all. This has been Conservation Science Today. I'm Tucker McPherson. Stay curious.